Okay, in the second line of defense, now we're going to talk about some antimicrobial proteins. One is interferons. Another one we'll talk about in a moment is complement. This is the way I want you to think about interferons. Of course, I want you to read on it. Think interferons are primarily against viruses. Interferons are primarily against viruses. Okay. So viral infected cells are activated to secrete interferons. Interferons protect the neighboring cells. So this is the way virus infects a cell among several cells, let's say. That cell is a sacrificial lamb. That cell is done for. But that cell will produce some interferons, which will then go to neighboring cells to make them be protected. That's the best way to say it. So let's look at that. So a virus enters a cell. The virus entering the cell then causes this cell, it's a sacrificial, to produce these things called interferons. The interferons then go to neighboring cells, sit on a receptor, and cause the neighboring cell to produce some antiviral proteins. And what it does is block the replication of the virus in the other cell. That's where it's like running interference in football. So then that cell, the virus cannot reproduce in the neighboring cells. So you can kind of look at that, just follow the sequence, and you can see what's happening. So interferons are produced by a variety of body cells. There are different kinds of interferons. Lymphocytes produce the gamma ones. They not only do what I said they would do with the viral, but they also stimulate macrophages to killer status, which can then eat the infected cell. Some, like white blood cells, produce alpha interferons, which reduce inflammation in the area. They still are going to do the viral thing. Fibroblasts, beta interferons also. So they are antiviral. In the cell being defended, the cell produces PKR. That means the neighboring cell. This blocks the virus from making a new protein in the whole cell. It blocks it at the ribosome. Remember protein synthesis? It can also reduce inflammation, so it's not a major process with inflammation in the area. So it tries to take it out quietly. Okay. We do now, biotechnology, have some genetically engineered interferons which can help in hepatitis and multiple sclerosis. That's applicational though. This particular man here is called a tree man. See, he's very susceptible because he doesn't have a good immune system to the papillomavirus, the one that causes warts. So he was genetically treated, I don't have his resultant pictured, and got rid of all these warts and everything. And you can glance on it. Papillomavirus can also cause cancer. Okay. We go next to the complement system. Now, I want you to think, where did the term come from? It's complementary to the antibody system, which we've not talked about. It's complementary to the antibody system. All right, this is an overview before we get into it, how it works. In your circulation, the liver makes some proteins. The liver makes some proteins. You have about 20 different proteins floating around the bloodstream unassembled. When something happens that needs the complement, it will almost be like Transformers, if you're familiar with that, or the old-time erector set, where all of a sudden these proteins will start clapping together and forming things. So initially they're floating independently, but when activated, they start snapping together to form the complement system. So they're floating independently. The complement system complements the antibody system, which we've not talked about. Okay? The complement system does three things, and I want you to think about the old Texas term OIL, O-I-L. The complement system does three things. in its effort to work. Opsonization, inflammation, and lysis. Let me say that again. The complement system, in its action to destroy stuff, does it by three methods: opsonization, inflammation, and lysis, oil. Okay? So, it amplifies the inflammatory response, kills bacteria, lysis, enhances opsonization. Remember, opsonization was to make tasty. Okay? There really are three pathways. One is the classical, the other is alternate pathway, and then there's a lectin pathway. I'm not going to get into the lectin, but I will mention the classical pathway and the alternate pathway. Okay. The way to activate complement, that means to activate the system to get it going, it has to bind something called IgG or IgM. Those are two antibodies. IgG and IgM are antibodies. So in order to activate through the classical pathway, there are three things that must what we call bind complement. There are three, and I'm going to show you the scheme in a moment. There are three things that must bind complement. Either an IgG, which is called immunoglobulin G, or an IgM called immunoglobulin M, or something called, I'll present to you in a moment, called CRP, which stands for C-reactive protein. So in order to get it started under the classical pathway, you either have to have an IgG bind to it, or an IgM, or CRP. Once that occurs, that sets the whole thing in motion. Now, setting it in motion, listen to me. This is what we call a cascade process, and you're going to see that in blood clotting too. Listen to my word, a cascade. Remember how one waterfall dumps into another waterfall that's larger, dumps into another one that's larger than that. So in many reactions in the body, we have a cascade, which means one thing is formed, and then that's formed goes and turns on the second thing, and then the result of that turns on the third thing, and it gets bigger as it goes downstream. It's called a cascade. You'll see that in the clotting process. All right. So each step, the preceder acts as an enzyme to get the next one going. So I'm going to go further now. This is kind of what you see. All right. The complement proteins, those have been numbered. So you have a C1, all of these are independent at first, a C1, a C4, and a C2. Okay? Basically what happens is something makes antibodies happen. We'll talk later. The antibody, if it's IgG or IgM or something called CRP, can hook to C1. C1's made by the liver is floating independently. When C1 gets bound by either IgG, we need two IgGs to do it, but don't worry about that, by either IgG or CRP or IgM, it will bind to C1. Once it binds to C1, then C1 can hook to C4 and C2. You have to bind first. These are separate. Once you bind, C1 hooks to C4 and C2 and forms this complex. This complex is going to act as an enzyme. So you're making an enzyme because you're making from C1, C4, and C2. The enzyme some will call C3 convertase. Some will call the enzyme C3 convertase, C-O-N-V-E-R-T-A-S-E, which is an enzyme. The C3 convertase then will come in and take C3, which is floating around, and split it. So this enzyme splits C3 into two fragments, a C3b and a C3a. The C3b can go and coat the microorganism that caused the whole thing, making it tasty. Opsonization. When that occurs, remember, neutrophils and our macrophages can come in and attack it. We've already talked about opsonization. That's the C3b. Remember, oil, opsonization. The C3a then can come in and stick to mast cells and basophils. Mast cells and basophils. They produce histamine, we've talked about. The histamine then can cause vasodilation, ultimately stimulating inflammation. Opsonization, inflammation. The C3b can also cause the splitting of C5. The C3b can also cause the splitting of C5 into a C5a and a C5b. The C5a can also, in addition to C3a, attach to mast cells and basophils, further enhancing inflammation. Then the C3b, 5b, 6, 7, 8, and 9 will form kind of a tight ring and stick directly on the microorganism's membrane. And that tight ring will actually pop a hole into the microorganism, which would then, the juice, the cytoplasmic stuff inside the microorganism will leak out, causing lysis. That's called a membrane attack complex. A membrane attack complex. So opsonization, inflammation, and lysis. Opsonization, inflammation, and lysis. Now, I took it from the classical pathway. The alternate pathway is that certain microorganisms, by themselves, without needing antibodies, can activate some of the 20 circulating proteins we mentioned, known as B, D, and P. That what will happen is, when that particular microorganism, based upon its cell membrane, can bind this B, D, and P without the antibodies of the C-reactive protein. When that occurs on that surface, then that complex here, with the B, D, and P coming together, can then act as an alternate form of the enzyme to split C3. Once C3 is split, everything occurs. So the key to complement is to split C3. The key to complement is to split C3. And once C3 is split, then you get all of the actions that you see. So that's how the complement occurs. C-reactive protein. I'm not going to say much on it. I don't know if it's even in your book this time. C-reactive protein is a protein made primarily in inflammation. It seems to have been an early attempt evolutionarily at the production of antibodies before antibodies came about. So it kind of works like an antibody, but it's too archaic. So it doesn't really do a lot for us. But it is of clinical importance. Listen to me. It is of clinical importance. Because what will happen is, if you get inflammation, we can look for it. Now, think about it. We already said inflammation. You can see redness and swelling and all that stuff like that. But you can't. That's only if it's on the skin. If it's inside the body, how you going to know it? So if the pancreas was inflamed or the liver, we wouldn't know it. But we could draw the CRP level. So it's an acute phase inflammation. Acute phase inflammation. Within 6 hours, it's going to rise if there's acute inflammation and peak at 48. So if we see it rising, we know we can confirm there's an internal inflammation. We have something called ESR also, and it comes in later. So these two things can be used clinically to determine if there's internal inflammation, CRP, and to do the ESR test. ESR is not a chemical, it's a test that's done. But CRP also can bind complement. All right, thank you.